Hello, everyone. We are pleased to have you join us for a presentation and live Q&A session. Dr. Emily Anderson, Principal Scientist with Revvity, will first present a talk titled Dharmacon CRISPR Guide RNA Design, the Key to Functional Knockout Every Time, and will then be joined for a live Q&A discussion with Associate Professor Kaylene Simpson, head of the Victorian Centre for Functional Genomics at the Peter MacCallum Cancer Centre. I'm Maren Mayer-Gross, and I'll be your moderator. Before we begin, I would like to remind everyone that this event is interactive. All attendees will be in listen-only mode for the session, but we encourage you to participate by submitting as many questions as you like at any time. To do so, simply type them into the question box on the screen and submit. We'll answer as many of the questions as we can during the discussion, where the topics will broaden to include applications from CRISPR guide design, high-throughput screening with RNAi and CRISPR, to future technologies and trends in the gene manipulation space. If there are questions that we cannot fit into the session, we will send an email response in the coming days to ensure your question has been answered. At the bottom of the screen, there are several interaction tools. All the tools are resizable and movable, so feel free to move them around to get the most out of your desktop space. You can expand the slide area or maximize it to full screen by clicking on the arrows in the top right corner. I would now like to introduce our speaker, Dr. Emily Anderson. A principal scientist at Revvity, Emily has worked to develop the Dharmacon reagents portfolio of gene modulation and engineering technologies, including on-target plus siRNA, Excel siRNA, and Editor, the first rationally designed genome-wide library of synthetic CRISPR guide RNAs. Emily received a PhD in biochemistry from CU Boulder, focused on the structure and function of telomeric protein single-strand DNA interactions. She has worked extensively on the mechanism, function, specificity, and delivery of RNAi. Inventor on several issued patents, she has also authored many peer-reviewed publications on RNAi and CRISPR-Cas9. Once again, thank you for your attention, and I will now pass you across to Dr. Anderson for the presentation.
Hello, I'd like to talk to you today about the importance of functionality and specificity in CRISPR guide RNA design, and how we can accelerate your work to achieve success in gene editing experiments. First, I'd like to give a brief introduction to Dharmacon reagents and the use of CRISPR-Cas9 for disrupting DNA sequence and knocking out genes in cell culture. Then we'll explore the logic and execution of a predictive design algorithm for guide RNAs. This was used for the synthesis of a genome-wide collection of highly functional and specific CRISPR guide RNAs. I'll cover some methods we used to validate and check functional protein depletion, as well as downstream phenotypic performance of editor guides. To conclude, we'll take a look at how these tools can facilitate gene function screening in a high-throughput manner and highlight the utility of complementary technologies to increase your confidence in gene function characterization. I'm Emily Anderson, a principal scientist at Horizon Discovery. I've worked with the Dharmacon reagents research and development team for 20 years, and have led many projects in the fields of both RNA interference and CRISPR gene editing, notably leading the development of the Dharmacon editor guide RNA algorithm, which is the focus of today's talk. Dharmacon reagents originated as Dharmacon, which for almost 30 years have been synonymous with high-quality chemical synthesis of RNA. This early expertise has inspired the creation of multiple product portfolios using RNA to manipulate and edit gene expression in cells. Dharmacon was the first to market for both algorithmically designed synthetic siRNAs and CRISPR RNA guides. We continue to grow and build upon these foundations to provide comprehensive solutions for gene modulation and editing, critical techniques for the development of medicines from small molecules all the way to gene and cell therapy. Central and core to all the reagents we provide is RNA synthesis. We make RNA. You could say that this is in our DNA, but we're actually talking about RNA, so pun intended. As I mentioned, we've been synthesizing RNA at the Lafayette, Colorado site for almost 30 years. This is located near Boulder, Colorado, considered by many to be the academic center of the RNA universe. It's an ISO-certified facility that excels in making quality custom RNA sequences, pushing the boundaries of length and offering a wide variety of scale and purification options. There's also a dizzying array of site-specific chemical modifications available, but that's a topic for a whole different webinar. Today, I'd like to focus on how our synthesis expertise, along with dedicated biology and bioinformatics teams, created a genome-wide collection of long synthetic guides for a particular purpose, CRISPR-Cas9 genome editing. Before I delve into the creation of the CRISPR-Cas9 editor guide RNA algorithm, I'd like to give a brief introduction to CRISPR-Cas9 and how it is used to knock out gene expression in cells. CRISPR is a bacterial immune defense system that stands for Clustered Regularly Interspaced Palindromic Repeats. Don't worry about the name. The main point is that this system has been harnessed as an RNA-guided DNA targeting technology. The CRISPR-associated nuclease Cas9 uses a portion of a guide RNA sequence to target a specific region of genomic DNA in cells through Watson-Crick base pairing. The target sequence regions are called protospacers, and they're located next to a short motif called a PAM, or protospacer-associated motif, which is the nucleotides NGG for the most widely used, Cas9. Upon successful targeting using a single guide RNA, or a two-part native CRISPR tracer RNA, Cas9 uses two nucleus domains to cut the DNA and make a double-stranded break. The cell can repair this break in several ways. Today, we'll focus on imperfect, non-homologous end joining, which creates an array of insertions and deletions, or indels, in the cells, leading to gene sequence disruption and ultimately knockout of the downstream gene product and its function in a heterogeneous manner. Jennifer Doudna and Emmanuelle Charpentier won the Nobel Prize in Chemistry in 2020 for discovery of this amazing system, which democratizes gene editing and promises to foster many breakthrough discoveries in biology and medicine. But how do you go from an elegant biological mechanism to a useful technology? For one, you need a source of guide RNA that will readily allow you to target your gene of interest. Warning, do not try this at home. Early on, when developing synthesis methodologies for these long synthetic RNAs, we tried an established method of producing RNA, in vitro transcription, or IVT. However, when transfecting this material into mammalian cells, we got spurious and variable results. IVT guide RNAs, but not chemically synthesized guides, either two-part native CRISPR tracers or single guide RNAs, stimulated the innate immune response and led to poor viability in several cell lines tested. Here's an example targeting two genes in HEC293 cells. This bar graph depicts qRT-PCR induction of the innate immune response genes IFIT1, IFIT-M1, OAS1, OAS2, and DDX58, which are potently induced by the IVT guides targeting both genes, as well as the poly-IC control sample. The chemically synthesized guides, whether two-part or single guide format, do not induce innate immunity. Therefore, we determined that synthetic RNA was ideally suited for editing in mammalian cells. To enable researchers to easily select one or several guides and guarantee a functional knockout outcome, we built a rational design algorithm with two core requirements: high function and high specificity for the chosen DNA target. Let's start with functionality. As I previously mentioned, it is important that a guide RNA should not only efficiently target, bind, and cleave DNA, but also that it produce a functional knockout. For this task, we used not high-throughput sequencing, but rather a high-throughput cellular reporter assay for gene disruption. This proteasome assay is a cell line containing stable expression of GFP, which is fused to an uncleavable ubiquitin moiety. When the proteasome pathway is intact, the GFP is rapidly degraded and there's low to no fluorescence. However, disruption of the proteasome pathway by chemical inhibition, knockdown, or here, knockout of a wide array of proteasome-associated genes causes visible and quantitative fluorescence, which is an excellent proxy for guide RNA knockout strength. We use this high-throughput reporter assay to test over 1,100 guide RNAs tiled over 10 proteasome genes. We transfected these and measured fluorescence as an indicator of guide RNA strength. A set of characteristics was then used to train a model that scored for function, weighting and combining features to best sort the poor-performing guide RNAs from the effective ones. When the predictions were tested using a leave-one-out model, high predictability was achieved with an area under the curve of 0.78 in the plot of true positive rate versus false positive rate. The model was tested and validated in various ways, which we'll discuss later. When distinguishing highly active guides from less active ones, we found that several expected features were important, like sequence preference by position, average GC content, and a strand preference. However, the algorithm uncovered more subtle features that added confidence to the selection, as well as distinguished combination effects between some of the features. Note that cut site location across the coding sequence of a gene, as shown on the right, has some effect, and that surprisingly, effective guides were found quite late in the coding sequence. Additionally, there's quite often a large distribution of guide RNA function within a given exon, shown in these circled regions. Some strategies for guide design, like biasing guides very early in a gene, shown by the arrows, as well as constraining guides to be close within an exon for easy sequencing of a set of guides, may not select the best guides. These picking strategies lose the advantage of selecting across a whole gene sequence and may force the selection of less functional guides simply due to their proximity. Another observation from this large dataset involved picking guides too early in a gene. We found some targets for which a selection of guides in the first exon, near to the start codon of a gene, caused efficient DNA disruption, as shown in this mismatch detection assay, but no functional consequence, as shown in the lower graph. While placement of early target sites might seem more likely to cause disruptive indels or stop codons leading to faulty mRNA, placing these too early is problematic when the gene uses an alternative start site, as do up to 15% of the genes in the Phantom database. In addition to highly efficient DNA disruption leading to potent protein knockout, a good guide must be specific. It should target the intended DNA sequence and avoid any other similar sequences in the genome, which could lead to off-target effects, muddying a phenotype outcome, or failing a promising therapy. We found that many public-facing alignment tools lacked the power and speed to identify all potential sites in the genome, not just mismatches, but also gaps, as seen in this figure. We developed an internal tool that exhaustively searches for all combinations of these flaws, both mismatched targets and gapped alignments. Additionally, flaws are assigned a weight by position as indicated by structural and functional studies of Cas9 binding, for instance, within and outside the characterized seed region. These specificity scores are combined with the functionality scores to pick the best guide candidates overall. This diagram highlights our design strategy, mapping it out on a hypothetical gene shown here. It demonstrates the importance of picking across a range of exons in your gene. What may seem to be a large array of NGG-PAM target sites to choose from can rapidly narrow down when both function and specificity are considered. So, for instance, CRISPR-A is quite functional, that has terrible predictive specificity, whereas CRISPR-B is a great choice and is both highly functional and highly specific. However, it is located in a smaller overlapping exon, where if you were forced to pick nearby CRISPRs, they might not be the best picks, or you'd pick CRISPR targets in the exon containing CRISPR-A and miss out on the most functional guide for this gene. This experiment shows the importance of an exhaustive alignment search. Since off-targets, including gaps, may not be searched by some alignment specificity tools, the search is incomplete and doesn't predict significant off-target risks. In this experimental example of 3 different types of 2-flaw alignments, we can see that sequence number 1, with 1 gap and 1 mismatch, does not function at all, as shown on the right in this T7 mismatch detection assay. A 2-gap alignment sequence, off-target number 3, also does not affect indels, whereas sequence number 2, with a gap and a mismatch alignment, is almost half as functional as the intended target, fully matched target in this example. Therefore, an alignment method that finds these alignments, including gaps, allows you to select guide RNAs that won't cut at these potential off-target sites. In addition, off-targets, including those that weren't predicted in the first place, are hard to dilute. Since the CRISPR mechanism for off-targeting is the same used for targeting, unlike siRNA and mRNA mechanisms, a poor specificity CRISPR loses its on-target strength equally with its off-target strength upon dilution, as shown in these 2 dose curves. These are consequential off-targets, with the relative indel formation shown at the top for the various 2 mismatch off-target sequences at the highest concentration measured. So, even at a low dose, selection for a high specificity CRISPR is crucial. Also, dilution of multiple CRISPRs, as in a pool, does not dilute out the effects of a poor specificity guide contained in that pool. It is best to have multiple independent candidates per gene to be confident in your knockout outcome. Now that we have an algorithm in hand to select the most functional and specific CRISPR guide RNAs for knockout, how do they fare in the real world? This is where extensive validation comes in. The first large-scale validation we performed used an amplicon sequencing assay by next-generation sequencing. But wait, before I tried to convince you that a functional assay for knockout produced the best results. In this case, we wanted to test that an algorithm based on data from a certain biological function was not biased to that particular function. For that end, we chose 10 high-scoring and 10 low-scoring guides across 10 genes of widely disparate function, location in the cells, etc. You can see from these charts that the high-scoring guides are much more likely than low-scoring guides to produce high levels of indel formation in the DNA. Over 90% of the picks in the high-scoring group perform better than 40% indel formation at their targets, whereas only about a third of the low-scoring guides, chosen essentially at random, perform at that level. This confirms that an algorithm trained on a specific biological function does well at picking guides generally, which have a high ability to target and cut DNA. In another example, we again tiled guides across a gene, in this case, the soluble tumor necrosis factor 1A. In an ELISA assay for detection of intact protein, you can see that the lower scoring guides on the left perform, again, essentially at random, with a large amount of poorly functional ones. While as the scores get higher to the right, these indeed are the most functional knockout guides for this protein, the function of which has nothing to do with proteasome degradation pathway. The editor algorithm picks functional guides generally, regardless of gene function in the cell. We've shown data supporting validation of the editor algorithm for its generality beyond the proteasome assay in which it was trained, in both disrupting DNA and causing protein knockout. Additionally, we've performed experiments to ensure that predesigned guides selected using the algorithm cause functional knockout reliably and consistently. In some cases, you will want to test the function of guides used in your specific experimental workflow. This is quite easy, as we provide primer sets to validate cutting and disruption at the DNA level by either TIDE or NGS. While you may use editor guides individually or pool them, as is often done in screening applications, it's best practice to transfect and measure these at the level of individual guides. The graph here indicates that for the editor reagents, which are placed in optimal positions across a gene for the highest function and precision, it is highly unlikely that a set of guides can be analyzed by one amplicon. Only about 20% of the guides are within 250 base pairs of each other across the genome-wide collection. Here I'd like to show some ways of measuring DNA editing and some of its consequences so that we're all on the same page interpreting these various data sets. When using CRISPR-Cas9 to knock out a gene, we are ultimately interested in editing at the functional level, knockout of the protein, or the phenotype that results from the missing protein, indicated in this experiment in blue. However, in the development and comparison of gene editing tools, often disruption at the DNA level is measured first. Indels can be measured with different methods: T7E1 mismatch assay, TIDE conventional sequencing, and NGS, or high-throughput next-generation sequencing. They indicate relative levels of efficiency specific to the technical limitations of the method used. NGS and TIDE typically report a higher percent of indels than a T7E1 assay, and there can be higher variability sometimes between technical replicates when using TIDE. Basically, when comparing guide sequence function, it is only apples to apples when using the same analysis method. Direct correlation of phenotype to sequencing methods like TIDE and NGS is quite good, but phenotype, in this case loss of a protein surface marker by FACS, is often much higher than a T7E result due to enzymatic limitations in that assay. Phenotype matters, which is why we'd used a high-throughput phenotypic assay to test guides for development of the algorithm. Here is an immunofluorescence experiment looking at the transferrin receptor protein. You can see that each individual selected guide and pooled together, whether delivered in a two-part or single guide format, achieves high editing efficiency as measured by TIDE and almost complete elimination of staining for the protein product. Although each reagent can affect the desired outcome, in a screening setting, it may be advantageous to pool guides and reduce the number of samples tested in a primary screen with use of the individual reagents in a secondary confirmatory experiment. Here's another example looking at the protein geminin. Note that in this example, by using immunofluorescence, you not only see the striking depletion of the target protein, but also the larger cell nuclei in the knockout samples, which is actually a phenotype of inhibition of geminin. Note also in this experiment that TIDE analysis can sometimes fail, is this missing data point, or lead to variable results, which is why you need the redundancy of multiple functional guides per target and to analyze them both by DNA and downstream protein or phenotypic analysis. Shown here are representative Western blot analyses of knockout of the proteins HDAC2 and AKT1. In this case, transfecting guide RNAs into a cell line that stably expresses Cas9. Again, with high confidence that disruption occurs at the DNA level, effectively leading to protein knockout, each individual guide results in 80 to 90% depletion of the target protein across the cell population in the sample. Predesigned editor guides were extensively tested, not only in adherent immortalized cell lines, but additionally in cell types that may be harder to manipulate, but are nonetheless very important in translational medicine, such as stem cells and primary immune cells. In this experiment, knockout of cell surface receptors by electroporation of a Cas9 ribonuclear protein was assessed by FACS, or cell sorting, in primary CD4 plus T cells. In agreement with the earlier data in cancer cell lines, functional guide RNAs in T cells resulted in high knockout of the target proteins, with the individual guides performing as well as the pool, again stressing the importance of good design for functional redundancy and the ability to follow up with individual sequences after using a pool. In the case of some proteins, strong and early phenotypes interfere with the analysis, as seen here with HLA-DR, where the edited cells may have been selected against before analysis. This is an application in induced pluripotent stem cells using Cas9 mRNA and synthetic sgRNAs targeting SOX2. One benefit of using mRNA and synthetic sgRNA, as well as ribonucleoprotein, is that the reagents are relatively short-lived to produce off-target effects, and they won't integrate into the cell's genome, as is a risk from plasmid-expressed reagents. Gene editing of SOX2 by tide analysis is shown on the left. All three guides generated greater than 80% indel formation while maintaining high viability of the cells. Functional knockout at the protein level is shown on the right. In the non-targeting control, the left column of images, pluripotency markers SSEA4, OCT4, SOX2, and TRA160 are all maintained in healthy stem cell colonies. In contrast, in the right column of images, where SOX2 is knocked out, not only do we see a loss of SOX2 protein, but also a reduction in other pluripotency factors, indicating that the knockout of SOX2 is strong enough to affect the maintenance of pluripotency in these cells. Keep in mind that with high knockout efficiency across the cell population, there's no need to isolate a clonal line to see a strong effect. This may not be true for every experimental phenotype, but if you start with potent and specific guides, you have a fighting chance. I'd like to take a few minutes now to go beyond knockout of individual genes and demonstrate how the ready availability of a genome-wide collection of knockout guide RNAs can further gene function research at a large scale. As a complement to the editor predesigned guide RNAs, we offer complete CRISPR gene editing solutions for both individual gene targets and for screening. Across the CRISPR knockout portfolio, we offer a variety of Cas nuclease and guide RNA options. Nucleases are available as lentivirus or DNA-free formats as either protein or mRNA, which can also be used with a fluorescent reporter for selection and enrichment of the transfected cells. We also offer a variety of ready-to-use Cas9 stable cell lines. Guide RNAs are available as modified synthetic sgRNAs or as lentivirus to suit your experimental needs. We also offer an all-in-one vector where Cas9 and guide RNA are introduced as a single lentivirus. All of our gene editing and gene modulation products are available in multiple screening formats, including pooled, pre-designed arrayed libraries, or custom arrayed libraries to suit a variety of applications. In an arrayed format, editor pre-designed synthetic guide RNAs enable functional characterization in complex assays. We tested editor pre-designed knockout guides in a high-content assay for cell cycle regulation. This work was published in a peer-reviewed article several years ago. This cell line expresses an EGFP tagged marker for cell cycle phase, which allows for high content analysis to determine changes in cell cycle progression and viability, as well as distinguish aberrant phenotypes such as multi-nucleation and irregular nuclei. To test the suitability of editor guides in eliciting phenotypes that can be distinguished by this high content assay, a library of guide RNAs was transfected into the reporter line, representing 169 genes with a reported role in cell cycle regulation. This example data is from a strong hit targeting aurora kinase B. Knockout exhibits a strong phenotype of multinuclear and irregular nucleus cells, consistent with this gene's product's biological role in the alignment and segregation of chromosomes during mitosis. Notice that all 4 CRISPR guides very strongly elicit this phenotype. Shown here are knockout phenotype heat maps of the CRISPR guides targeting many of the cell cycle-related genes in this study. You can see that knockout of many of these genes results in robust phenotypes with multiple guides confirming the effect. And there's a clear distinction between gene targets with different high-content phenotypic outcomes. The relative absence of hits with only one guide per gene suggests that these CRISPR guides are highly specific and are not generally prone to off-targeting that would obscure interpretation of this high content result. Since their introduction, editor guides have been used in many gene function studies, both for detailed pathway analysis and in high throughput screening contexts. Here are just a few recent examples highlighting HIV function in primary T cells, immune cell co-culture for cancer killing, histone deposition, and chromosome instability, EGFR and P53 pathway interaction, regulatory T cell control networks, and the innate immune response in coronavirus infection. To learn more about the role of editor guides in these great studies and others, scan this code for a larger editor reading list. Hopefully, you've gained useful insight into the use of CRISPR-Cas9 for gene knockout and the importance of using highly functional and specific targeting guides. I'd like to conclude by telling you a little bit about methods you can use beyond CRISPR knockout to strengthen the results of your gene function studies. We've gone over how using the targeting and cutting ability of CRISPR-Cas9 can be harnessed for DNA disruption and knockout at the gene sequence level. However, depending on your needs, we provide solutions for multiple technologies that complement each other and strengthen the outcomes of your loss of function studies. In addition to editor knockout guides, you can utilize CRISPR inhibition guides with our proprietary and potent repressor domains, which knock down gene expression at the transcriptional level with nuclease deactivated Cas9 and gene promoter targeting. And to come full circle, I'll remind you that Dharmacon reagents are renowned for their RNA interference reagents, which knock down expression at the post-transcriptional level by targeted mRNA degradation. As an example, although stay tuned for later in the year when there'll be another webinar exclusively focused on this topic, here are quantitative results of protein knockdown targeting 3 genes in the epithelial mesenchymal transition pathway after gene expression reduction by 3 methods: siRNA, CRISPR knockout, and CRISPR interference. While the results are similar, each of these tools affects expression reduction in a different manner, such that confidence can be gained in the phenotypic outcome by using orthogonal methods of interrogation. Here is our last example from the earlier RAID screen setup of cell cycle outcomes. We are comparing the high content phenotypes of knockout CRISPR RNA guides to the use of siRNAs to knock down the same genes. In this case, for highly functional guides and robust phenotypes, you can see that largely there's agreement between the 2 techniques. Only the one instance on the far right, the CDKN1A target, is discordant, where further studies would be needed to elucidate the role of this gene in this assay. The message here is redundancy. Sets of individual reagents increase confidence, as does ready access to multiple toolsets to strengthen outcomes. To conclude, I hope that I've convinced you that Dharmacon reagents are provided by a trusted partner, from RNA synthesis to proven solutions to regulate gene function. Optimal CRISPR knockout guides are both highly functional and highly precise, ensuring that you achieve the best outcome not only for DNA disruption, but for downstream protein knockout and robust, clean phenotypes. Whatever method or combination of methods you use to study gene function in cells, we can help you achieve the best results so that you can focus with confidence on what's next in your research. Thank you to the biology research and development team, bioinformatics, chemistry and manufacturing teams, and everyone involved in the development of the editor portfolio.
Hello and welcome back, everyone. We are now ready to start the live Q&A session where Emily Anderson will now be joined by world-renowned screening leader, Associate Professor Kaylene J. Simpson, head of the Victorian Centre for Functional Genomics at the Peter MacCallum Cancer Centre, enabling researchers Australia-wide to perform high-throughput target discovery using gene targeting approaches such as CRISPR-Cas9, RNAi, and broad-scale compound screening in both 2D and 3D settings. Before we kick it off, I would like to remind everyone that you may still submit questions into the question box on your screen, and we'll get to as many as we can. The session will be organized loosely around a few topics, so please, ask away. First, of course, any questions directly related to Emily's presentation content, happy to get those in the session. Next, any questions around arrayed or pooled RNAi CRISPR screening, where Kaylene can share her vast, knowledge. And finally, any general questions about gene editing or gene modulation technologies or future perspectives for the field. Let's see, here we go. Let's see some of the questions that we have in the queue already. The first one that I see. Okay, this one's probably for Emily. How is your algorithm different from other resources? And yeah, go ahead, Emily. You can go ahead and answer this one.
Thanks, Marian, and yeah, thanks everybody for attending. So there's plenty of resources now. CRISPR's been around for a few years that claim to help with CRISPR guide design. And oftentimes when people say design, they mean looking at a gene sequence and looking at PAM site identification. So for pyogenes, CRISPR, it's an NGG site. And they'll show you all the sites you can target, but they don't really help you select a functional guide. They're just showing PAM sites. Also, there are other vendors, some vendors claim to have an algorithm that they've trained on an NGS-based cutting assay. And so, we showed data on that for the validation, but cutting doesn't always result in a knockout or protein depletion, which, we also showed. So I'd say the main difference is our algorithm was informed and tested on phenotypic assays to ensure that the guide that targets and cuts actually gives a knockout. And so one of the benefits of this approach is that it creates the stronger phenotype across the population of cells. And in that case, you may not even need to do clonal isolates. You'll have high enough knockout in the population for a lot of targets and a lot of downstream applications.
All right, thank you, Emily. That's great. Okay, let's see here. The next question that I see in the queue is, what are the challenges setting up a screen that researchers potentially face with their RNAi or CRISPR screening experiments? This one should go to Kaylene.
Okay, thanks. Nice to be part of this terrific conversation. I suppose my lab has run RNA interference screens for the last 15 years. And as we progressed our way into CRISPR and synthetic CRISPR, there was a number of little hurdles that we had to get over. Primarily, we work with high expressing Cas9 cells. So we've shown that if we select the top 20% Cas9 expression, we will get the best editing we can. And everything we do in an arrayed screening format depends on your controls and your high-level transfection efficiency. So the one thing that we spend a lot of time on is getting that high transfection efficiency so we can get at as best possible editing in every cell. We recognize that we can't get editing in every cell, not necessarily. So then when we are looking at our readout, it's going to depend on the type of readout that we use. We do a lot of imaging, high content imaging, and in that setting, if we need to, we can proceed to single cell analyses that will allow us to more specifically identify the populations that have had editing.
All right, thank you. Let's see, the next question in the queue, looks like this one's for Emily. How many CRISPR guides should I use to knock out a gene? Yeah, and this seems to be not screening related, probably just experimental.
Right, so if you're interested in a single very specific gene for knockout, and as Kaylene alluded to, sometimes for cell line generation, clonal selection, we recommend about 3 guide RNAs. And so that's how many pre-designs are available, readily offered on our website. And I would also say, specifically for cell line generation, we would recommend testing them individually for knockout and proceeding to generate a cell line individually. This allows you to confirm the best performer, so you get the best knockout and you don't have to sift through as many clones. And testing 2 or 3 also allows functional redundancy, which you can be reassured in the results because you've knocked out that gene in multiple locations.
Thank you, Emily. Let's see, the next one in the queue is, yeah, I think this one's probably for Kaylene. Which areas are we still struggling to address, and how does screening need to evolve to address current roadblocks?
Excellent question. Thanks for that one. For us at the moment, the area that we really want to be able to forge into is immunology. So within our institute, there is a massive emphasis on immunology as therapy, and we've been trying to do these types of screens that synthetic CRISPR has really enabled researchers the capacity to do single target work where we use nucleofection as a means by delivering the guides into suspension cells. The challenge for us is, though, that this still is a really expensive exercise in a high throughput manner, and certainly when we use nucleofection at a 96-well format, it is now doable, but we still need a lot of guide, and we can't do this at the sort of scale that we would traditionally do synthetic screening in a 3 to 4 well format in. So for us, this is an area we're spending a lot of time in, and I hear people might think that it's possible to do these type of pooled screens with viral vectors. We do this as well, but it would be ideal if we could do an arrayed, very simple method of direct delivery of the synthetic guide to suspension cells. That would make a massive difference. And whilst it's possible, it's not at the level that we can do it on scale of any other 2D types of experiments. And the other thing I would add is our primary interest in our lab at the moment is developing the tools to deliver gene targeting to cells growing in a 3-dimensional matrix. So matrigel embedded patient-derived materials where we can actually directly gene edit. And that's something that we are working heavily on, but it's something we don't have a solution to, but is the direction we can see us going.
Oh yeah, that's fascinating. I'm excited. I hope we get there soon. Let's see, actually there's another one that came in for you, Kaylene, that I just came across, and it is, where do you see high throughput CRISPR screening moving in the next 5 years? Does this include CRISPR-A and CRISPR-I libraries?
Absolutely does, actually. We've owned these libraries as pooled libraries now for a number of years, but we haven't had anyone do too much in this space, and I can say that it's just starting to become of great interest. And for us, given that we've got a lot of synthetic emphasis in our lab and the capacity to do arrayed individual well screening, we can see now that people will be coupling knockout screens and arrayed screens, and overexpression screens in particular, together to get a more detailed viewpoint of the genetic outcomes when you knock targets down or overexpress them. Whether they are concomitantly opposite, we don't know. We suspect most likely not, actually, but this will give us the better, broader picture of how to do that.
Great, thank you. Let's see. The next one that I'm seeing here is, this looks like it maybe is a good one for Emily. Are there advantages to using pooled guides over individual guides?
Okay, so I'm assuming in that context you mean in an arrayed format, pooling guides, same gene in one well.
That's what it sounds like they're trying to ask here. Yep.
Right. Okay. So not pooled viral. So I would say, are there advantages? Yes and no. So sometimes in high throughput functional genomics screening at very high levels of genes, like Kaylene's lab does, pooling multiple guides per gene, even CRISPRs, is helpful to reduce reagent costs. And this is a standard practice and has been for decades with siRNAs. And in this context, we've done experimentation with it, and pooling has generally not shown to have a detrimental effect on the cell population, pooling a few guides against a gene. And again, if you measure phenotype in this heterogeneous population in a well for a given gene target, you're not going to go through and sequence individual indels and cells. You're doing it in a population. So for primary screens, that can be a good tactic to reduce the plates and the number of reagents you need. However, we don't really recommend pooling sequences outside of the high throughput screening context, probably for a couple of reasons. The first would be analysis of the edits can be complicated and difficult or impossible to even interpret when you're cutting in multiple locations. And second, we've seen that using multiple guides in the same genomic region, while you might be able to cut out, per se, a section of the gene, like do true deletion, in a very small percentage of the cells, 1%, plus or minus, you can see large, like, chromosomal rearrangements. And these are also sort of hard to identify at that low frequency, and could cause changes not due to the knockout of that single gene. So pooling is useful in sort of primary high throughput screens. I wouldn't recommend pooling guides to target assessing a single gene and when you want to isolate, let's say, clones for knockout.
Thank you, Emily.
I was just going to pop in to add that, on the scale when we do a genome-wide screen, it's a massive exercise. So pooling does make 100% sense at that level. But we will always validate our screens at the individual guide level. So breaking that pool down into its 3 subunits will give us the highest confidence outcome that we can identify the most robust hits from that. So if only one guide gave us an outcome and 2 other guides didn't, highly unlikely, but if that's the case, we would be less likely to follow that target up as a hit.
Yep, so that's what you're saying, that functional redundancy is important in individual wells. And I also forgot to mention that we have the same sequences for gene targets available both as single and as pooled. So you can set this up either way.
Excellent. Okay, let's see. There's another one here. It looks like sort of a technical instrument question about screens. So Kaylene, I think this one's for you. What technical and resource instrument requirements are different between a smaller scale experiment or screen with just like, they're saying tens or dozens of genes, and a much larger screen such as the whole genome ones you were mentioning?
Yeah, when I saw this question pop into the chat, I was thinking, tens or dozens might sound like a lot to someone, and it's certainly not on the scale of us. I used to make jokes that we wouldn't get out of bed for a less than 1 384-well plate. But there's 2 ways to answer this. We have a huge amount of automation and we have highly skilled people in our lab that enables us to do every step of the screen in an automated fashion. We do quite a bit of optimization manually, just because it's a little bit faster sometimes if you've just got very small scale work to do. And it's feasible that if you can prepare it in a 384-well plate, you can do that. But what happens at the end of the day is that you will always have more robust outcomes if you can do some level of automation. So if you don't have access to it and you're doing 20 or 30 targets, it is a little bit more effort, I guess, to do that, but it's doable. But we would always suggest that beyond the optimization manually, even if your screen is small, automation does help you because you will get more robustness and you will be able to do more replicates just by the nature of you don't have to pipette anything yourself.
Great. Thank you. Let's see. Okay, there's another one coming in here. It says, thank you for the excellent presentation, Emily. I have a question regarding guide RNA for knockout. Is there a relationship between the efficiency of editing with selected guide RNAs and the induction of random indels across the genome?
Okay, so if I understand you correctly, so many of the early publications and beyond looking at guide RNA specificity, there's been a couple studies, whole genome sequencing of clones. There's been many studies with these more targeted methods, guide-seq, circle-seq, site-seq. And most of the off-targeting, so I'd say for a specific guide that you assure bioinformatically that the sequence does not have low amounts of mismatches in the genome, it's hard to detect off-targets. And the vast majority of ones that you find when you're looking for them are actually not, I would say, random across the genome. They can actually be attributed to this sequence mismatching, which, like I said, if you have good bioinformatic design, you can find a knockout guide that's designed to avoid that. You know, the first couple of studies that came out doing whole genome sequencing on clones, there were these back-to-back papers. They did find a few random edits in the genome that, by their statistical analysis, were actually sort of drift in genomic sequences as the cells divide, right? I mean, the genome is not absolutely static and homogeneous through time. So I would say, you know, with the CRISPR technology, there's very low amounts of, you know, random editing, and then it's not really attributed to the CRISPR, it's just genetic drift. So I would say it is possible, and there are CRISPR sequences in clinical trials even, to pick very highly selective sequences that are selective through the CRISPR mechanism and that have high efficiency for the on-target and then do not show sequence-specific off-targeting.
Thank you, Emily. Great answer. Let's see, there's another one here. I think this one, probably Kaylene. This says, thank you for such a great talk. Do you have any insights, future perspectives on siRNA for selective gene knockdown for cancer treatment?
Well, you've hit me where my passion was for 20 years of doing RNAi.
Right?
And it's funny that when I lecture now to young scientists, most haven't heard of RNAi. So it's still there and it still exists. And obviously there's been some very clear therapeutic strategies and companies that have used RNA interference. Whether RNAi will be completely usurped by CRISPR-I, I don't know. That's what we're going to be looking at in the future. But I will say that we still do use RNAi a lot in our lab. We use it as a tool to develop assays, techniques that we want to be able to pass on to our end users. And the reason is it's easy to use. It's almost more forgiving in a transfection setting than CRISPR is, so you don't have to do as much work to get that optimized. But it means you can, if you don't have your Cas9 expressing cells, you can develop your assay, get your readout working, and then transpose all of that to a CRISPR setting. Or you may want to triage your target list down a little further, or you may want to validate your CRISPRs. So there's all still different utilities for it. I think sometimes community of scientists see a new technique and think that's the one that they should be writing in their grants. And we always say that your technique has to be fit for your purpose. So I think RNAi still has some place at the moment.
Excellent. We, of course, like to hear that too.
And I also, you know, Kaylene,
yeah, Emily.
At delivery, you know, what she's saying about transfection efficiency and optimization in screens, if you think you're delivering this to an adult, you know, or a human as a therapy, delivery is even more of, say, a challenge. So there's definitely, I mean, there are approved siRNA drug therapies. Granted, the technique is older, but it's just partly a reflection of delivery, and that for the CRISPR system, you're going to have to get a protein complex or a mixture of messenger RNA, small RNA, you know, into the human body to your target organ to get, you know, so definitely the delivery challenge is different, right, with RNAi and with CRISPR.
And Emily, I'll just add to that, because I suppose if I relate the strategy of understanding a gene target's function by suppression, that is typically what we do in a cancer setting when we treat a patient with a drug. So there is always going to be a disconnect if we've got a knockout strategy to relate to a therapeutic strategy. So this is a very multifaceted question, and there's not really an actual answer, but it provokes a lot of conversation because these different techniques exist. So it's just worth thinking broadly.
And that's why it's, before the question about the future of screening, including techniques like CRISPR-I, and like you said, correlating CRISPR-I to CRISPR-A or seeing how those interplay, is suppression, similar different to knockout. And then there's even kind of what people may call these third generation techniques like base editing, prime editing, that also can manipulate the sequence of a gene in a different way. So I think it's really exciting to imagine the interplay for the drug target discovery and therapy that all these techniques are going to bring.
All right, absolutely. And I think actually we have a couple more questions here. I think we probably only have time for one more. And there's one actually that just popped in. It's we'll see if we can get this one. A technical question. Any considerations moving from cells to the in vivo context to explore screening hits in mouse or zebrafish? And then does Horizon support in vivo experimental research? So I guess maybe both of you could answer that, but the in vivo question for Emily, which I think we know. Yeah.
So as far as like the guide RNAs, at the Dharmacon site, we make everything from small scale. I think I mentioned at the beginning of the presentation, up to tens of grams or more, very large scale, and there are various synthesis scales and purity options that would include supporting at least mouse and kind of preclinical in vivo studies, for sure. Again, with the consideration of delivery, and do you want to use a ribonucleoprotein? Are you going to deliver the Cas through mRNA plus guide? I mean, in vivo studies are a little more, there's more to consider, but yes, we do support that research.
Great. And then, yeah, I just want to give you both one last chance to share your thoughts on future exciting technology. We kind of just talked about that, but if there was anything else you wanted to just also include before we wrap up the session, since we're at the top of the hour here.
I mean, Kaylene, we've already discussed, the exciting frontiers with CRISPR modulation, interference, activation, base editing, stuff like that. Are there other things you'd like to, in the screening world, that you're excited about, Kaylene?
We're very much focusing on multi-omics strategies right now. So integrating CRISPR screens with drug screens and being able to get a whole transcriptome sequence in a 384-well plate that can allow us to look at on-target, off-target specificity of any screening, so we can get through validations very quickly, but we can also look at other perturbations. Obviously, there is Perturb-seq and other types of strategies that are coming out as well. And there's clearly a lot of activity in labs using other Cas nucleases. So there's a huge amount coming on in the future, I think.
All right, well, thank you both so much for the engaging conversation and the presentation. And thank you for the audience for giving us such great questions. If we didn't have time for your question, we'll make sure to reach out to you in the next 24 to 48 hours with answers. And this webinar will be available on demand at the same URL within the next day. So you can rewatch anytime or share with a colleague that wasn't able to attend today. Thank you so much for joining us, and have a wonderful rest of your day. Thank you. Bye-bye.